first in what will be at least 2 parts on 3.4 inheritance. And this is going to go over simple inheritance, the dominant and recessive type of inheritance that was discovered by Gregor Mendel. Gregor Mendel was actually a monk, and so he had a lot of quiet time on his hands for thought and reflection, which is super important for creative thinking in science. And he had a garden. Well, he noticed that the pea plants in his garden were doing some interesting things with respect to what was being passed on in different generations, and this spurred his curiosity. So see that you can really see the scientific process here. The observation spurred the curiosity or the question, and then he set out to answer that question in a very methodical way. He used a very methodical approach. Pea plants turned out to be excellent for making this type of discovery because they grow quickly. They have structures that are readily identifiable and observable. It's not like he had to have any fancy equipment in order to see the traits in the pea plants. And they have very distinctive dominant and recessive traits, which means they have this, what we characterize as simple inheritance pattern for many of their traits. Some important terms that we need to note and review as we move into a discussion of how traits are inherited. Our gene. Now, if you remember, the gene is a segment of DNA that codes for a particular trait. And one thing that isn't mentioned here that should be is locus. A locus is the position of a gene. It's the particular position of a gene on a homologous chromosome. So the particular position of a gene on a chromosome, and that position is the same on homologous chromosomes. So for example, if this was from the male part of the plant, and this is from the female part of the plant, so this would be equivalent to mom, and this would be dad. You can see these are homologous chromosomes, so the gene for flower color is at the same locus in both cases, but the allele is different. Here, the allele is for purple petals, and here the allele is for white petals. The flower color gene, again, same locus on homologous chromosomes, but the specific variant of the trait or allele is different. Okay, so next is genotype. Genotype refers to the genetic makeup. So what specific combination of alleles does an individual have? That's their genotype. Genotype codes for phenotype. Phenotype, you can remember the PH at the beginning of phenotype, phenotype physical. So the phenotype is associated with the physical characteristics. So, for example, what color is the flower actually? Is a person tall or short? What's their hair color? So that would be the phenotype. And going back to what we have been discussing, well, really since Christmas and a little before, the central dogma, remember, is DNA to RNA to protein. And we've looked at that in great detail. And so in this case, the DNA, that's your genotype. And then what actually gets expressed is the phenotype. So the genotype is an individual's particular combination of DNA or alleles. And the phenotype then would be whatever proteins are being expressed to generate the physical characteristics that you see when you're looking at an individual organism. So Mendel observed 2 types of traits. He saw dominant traits, and in the case of a dominant trait, this is the one that you're going to see in the phenotype. So whether there are 2 dominant alleles present or just 1 dominant allele present, in the case of simple dominant recessive inheritance, the one dominant allele is all that's needed in order for that to be the phenotype. Dominant alleles are shown with a capital letter. So, for example, here, the capital B is for brown eyes. In that case, the recessive allele, which is shown down at the bottom, is blue eyes. And that brings us to recessive alleles. Recessive alleles can be hidden or masked by the presence of a dominant allele. In order for a recessive allele to actually determine the phenotype or the physical characteristic, 2 recessive alleles have to be present. And they are represented in lowercase letters. So using these designations with capital and lowercase letters, we're able to tell when we look at the genotype of an organism, this is the way it's represented. and the letters, we're able to tell whether it's homozygous or heterozygous. That prefix homo means the same. So a genotype is considered homozygous if both alleles from both mom and dad, are either dominant or recessive. So if both alleles are dominant, it's homozygous dominant, and that's what you see here. If both alleles are recessive, it's homozygous recessive, and that's what you see here. The word heterozygous, hetero, is opposite or different. Heterozygous means that the alleles from the parents are different, meaning one, in the case of simple inheritance, one is dominant and the other is recessive. And so that's what you see here with a heterozygote. Sometimes you will hear the terms purebred and hybrid used when referring to organisms with a particular genotype. An organism may be categorized as purebred if both of the alleles are the same. So an organism that is homozygous dominant or homozygous recessive is considered purebred, while an organism that has one dominant allele and one recessive allele would be considered a mix or a hybrid. You'll also hear the term carrier used frequently. A carrier is an individual that's heterozygous for a gene, but because they're heterozygous, they're showing the dominant phenotype, but they're still a carrier, meaning they have that recessive allele that they can pass on to their offspring. Most often, you'll hear the term carrier used when talking about disease, genes or alleles associated with disease, but it is still correct terminology, even when speaking of a normal recessive allele, the term carrier. Now, I talk a lot about how important it is to connect what you're learning to what you already understand. We just covered meiosis, which is shown in the schematic here. You see the diploid cell at the top of the diagram here. And then there are two different genes highlighted, R and Y. And so gene R has the locus, let's see if we can move in here. Gene R has the locus here. And so you can tell that these two in this case are homologous. And then you can see the Y locus here on this different pair of homologous chromosomes. So these would be homologous. And then what you see here is metaphase 1. They're just showing two different options here. So based on independent assortment or arrangement, you can have different arrangements of the chromosome pairs during metaphase 1. So here's one possibility. Here is another possibility. So don't let that confuse you. It's still just regular metaphase 1. They're just showing how these different ways of aligning at the equator during metaphase 1 can lead to the generation of different gametes. So in metaphase 1, the homologous chromosome pairs line up along the equator. And it's even easier to see now how the genes are at the same loci on each chromosome in a homologous pair, but the alleles are different. You know the alleles are different because they're represented in uppercase and lowercase letters. So in the case of both of these genes, in this example, the alleles are different. You then see meiosis 2, which involves the separation of sister chromatids, and ultimately you end up with 4 possible gametes. Now remember, you're just looking at 2 examples here, so it's still an outcome of 4. And I just wanted to review this with you because when you set up a Punnett square, when you look to predict simple inheritance, you're really working within your understanding of how gametes are generated within an organism and ultimately how those gametes combine to form a zygote and pass along that genetic information. So it's super important that you continue to connect it all together. Okay, so just continuing with that, here you've got 1N haploid gametes, and they are combining to form a 2N diploid zygote, which is the very beginning of a new organism. A zygote is a fertilized egg. So the example here is human beings because we have 23 chromosomes in each gamete combining to form a total of 46. And if you look here, you can see that they are in 23 homologous pairs. You can also tell that this one is, hopefully you can tell by looking at it, that it's a male because showing the Y chromosome smaller. So this is everything that we're doing now is very connected to this meiosis. So these homologous pairs, this is the different possibilities for a gene at a particular loci. So a homologous pair may be homozygous dominant, meaning that the allele for a particular gene is dominant in both the chromosome from mom and the chromosome from dad. It could be heterozygous, dominant in the case of mom, recessive in the case of dad, or recessive in the case of mom and dominant from dad. It could also be homozygous recessive, meaning that both mom and dad delivered a recessive allele for that particular gene in the gamete. A Punnett square or Punnett grid, you'll hear each of those terms used. It's the same thing, just different ways of saying it, is used to show how the alleles of parents are split between their gametes and how new combination of alleles can show up in their offspring. So it's a four-part square, and it allows the determination of predicted genotype and phenotype ratios. So again, connecting back, if these are the female gametes, so if mom is represented up here, then these would be in eggs, right? So these are the different possibilities. If mom was heterozygous, then in the egg that is going to fertilize the zygote, there could be either a dominant allele or a recessive allele. These would be from dad then. So I'll draw that to represent sperm. And again, you don't have to do this every time. Just don't disconnect from the entire process here. Remember that what's represented when you draw a Punnett square like this is the gametes from mom and from dad. So in this case, dad was also heterozygous. So there's a 50% chance that he's going to send the allele, the dominant allele for this trait in a gamete, or the recessive allele. So once you've set up the parents' gametes like this, then you are just combining them together in the square. So this F is here, this one is there. So since we've got two dominant, it's FF here and here, here and here. And as you can see, the capital letter is always first. If there is a dominant allele, it's always represented first. And here and here. Now, once we've done this, we can predict both the genotype and the phenotype. The genotype is the individual letters themselves. So, for example, the genotype in this case is homozygous dominant. The genotype in this case is heterozygous. Heterozygous here, homozygous recessive here. The phenotype, in this case, the trait is freckles, and it's designated dominant in this example. So the phenotype would be, in this case, freckles, in this case, freckles, in this case, freckles, and in this case, freckles. So our genotype ratio is going to be 1 to 2 to 1. Homozygous dominant, heterozygous, homozygous recessive. But our phenotype ratio is going to be 3 to 1, freckles to no freckles. All right, let's practice some more. So in this case, this is an example from Mendel's work, and it's pea plant height. So he said each parent, or it says here, each parent was purebred, one tall and one short. Remember, purebred means homozygous. So if the tall genotype is dominant, then that one parent would have been homozygous dominant, which is what you see here. The other parent, homozygous recessive, which is what you see here. So we set that up and see what our results are. Now remember, we combine what's above and to the side. All right, so all of my genotypes are heterozygous, and all of the phenotypes, since tall is dominant, and anytime the dominant allele is present, you're going to see the dominant phenotype. So since tall is dominant, the phenotype is tall. The genotype ratio is 0 to 4 to 0, because there are 4 instances of heterozygotes and no instances of homozygous dominant or homozygous recessive. And this is considered the first filial generation, or F1 generation. So when you're setting up this type of work, the purebred parents are considered the P, or parent generation. The first generation of offspring is considered the first filial generation. And so to Mendel, this was really interesting because he had a short plant to begin with, and now the short trait has sort of disappeared. And so that led him to think, is it gone forever? Where is the short trait? And so then he crossed the first filial generation and looked at what happened in the second filial generation. So remember, these are both F1, because all the F1 offspring were heterozygous. And so what would happen if he crossed the female gametes from an F1 plant with the male gametes from a different F1 plant? Well, this is what his results looked like. So the genotype ratio here is 1 to 2 to 1, because there's one homozygous recessive, 2 heterozygotes, and 1 homozygous dominant. Sorry, I said this backwards, but you can follow. The phenotype ratio is, let's see, this one's going to be tall because there's dominant. This one's going to be tall. This one's going to be tall. And this one, because there's 2 recessive alleles, is going to be short. So we have a 3-to-1 phenotype ratio of tall to short. Now, once you understand how these squares work, you can use it to work backwards and figure out what's going on with a parental plant. So, for example, if there's a recessive phenotype, you only have one possibility for the genotype. So you don't have to, if you know that a particular trait's recessive and you see that that organism is showing the phenotype for that recessive trait, then you already know that the genotype is going to be recessive. And I'll just use this example has p's, so I'll just use p's. So these are lowercase p's. I'll draw the line so you can tell they're lowercase. So if it's got a recessive phenotype, you already know for sure that it has a recessive genotype. But what about in the case of a dominant phenotype? Dominant phenotype, forgot a letter there, could be either big P, little p, or big P, big P. And if you don't know for sure, the way that you can figure that out, if you don't want to isolate the genes and run a gel and do that type of analysis, is by crossing them with a recessive genotype, a known genotype. You really could, if you knew for sure that another plant was homozygous dominant, that would work too. But usually recessive genotype is used in a test cross because you know just by looking at the organism that it is indeed recessive for both alleles for that particular gene. So if you're trying to figure it out, and that's what's shown here, you can predict what would happen in each case if it's homozygous dominant or heterozygous. And then, based on the offspring and the ratios of the phenotypes in the offspring, you can determine whether or not the parent plant was heterozygous or homozygous dominant, as long as you have enough offspring to do so. So if the parent plant in question is homozygous dominant, then when it's crossed with a recessive phenotype, all of the offspring are going to show the dominant phenotype because they're all going to be heterozygous, as you see here. However, if the parent plant is heterozygous, then you're going to end up with 50% dominant phenotype and 50% recessive phenotype. And so test crosses are used in that way to work backwards and determine what the phenotype of a particular plant is in the case that it, or what the genotype is in the case that it's a dominant phenotype and you don't know whether it's homozygous dominant or heterozygous. All right, some next steps for you. I have some SpongeBob genetics practice. It's 2 worksheets front and back. You may zip right through it. My intention is not to waste your time. But the good news is if you already know how to do it, it's not going to take a lot of your time. I just want to be sure, without knowing how much you all retained and what everyone's background is, I just want to make sure that we were on the same page before we move into some of the more complex types of inheritance and add that kind of IB level complexity to things. And then once you've done that, there is a slideshow. Well, it's PDFs, but I think it's called slides, but I converted it to a PDF to put it into course materials so you can look at those slides to check your work. Again, the purpose of it is practice, but you do want to go through at least the majority of it, at least work through the problems and a little bit of each of the exercises to make sure that we are all on the same page and you're comfortable with this information. Once you've done that, there's going to be TED-Ed and Study.com that you can work on. If you have any questions, just send me a Schoology message or an email, or talk to me face-to-face in class or during H2H. Thanks.